The focus of this project is on precancer, and what that means is there are some changes in the epithelium that we can often see already on a clinical basis. So let's say a patient comes to a dentist or a specialist, dental specialist, and the clinician finds an area that's not normal, and they'll often do either an excision or take part of that lesion and submit it to someone like me, a neuromaxillofacial pathologist, for evaluation under the microscope. I do the diagnosis, and I use microscopic features that help us arrive at the most accurate possible diagnosis. Now, the problem is that even doing that is generally not good enough. What we see clinically and under the microscope doesn't tell us, A, how severe the problem is sometimes, and many times we can't tell the patient the probability of this becoming a cancer is 50%, 90%. We can't do that. And because of that, we need other ways to study it. Now, Dr. Cui has had a very sophisticated approach to studying cancer, including the cancer cells and the surrounding cells, immune system cells, cells that are important for stroma, in specimens. And this is a computer-based digital analysis that allows to study some of those interactions. But this new method that's now become available, and it's called CosMx.
It's really an unbiased approach. So we can use 6,000 genes, okay, the RNA probes for 6,000 genes. Basically, we can scan through the whole human genome and identify what's changed or in which specific cells, and also in the spatial context. So that will assist us to understand how cell-cell interaction plays a role in the precancer development or the precancer progression. That will help us for the diagnosis and prognosis.
So when this new technology became available and Georgia Cancer Center acquired it, we got very excited because now we can use probes that tell us which genes are expressed in every single cell in that environment. Moreover, Dr. Cui has a way of analyzing, making sure that the cells that are interacting are close enough together that tells us, yes, indeed, there is an interaction. And by looking at which genes are expressed, we can tell which cells are talking to each other and what the language is. What is that conversation? Because we know that it takes more than just the epithelial cells to become cancerous. It takes a very nurturing environment to push the transformation along to become a cancer, okay? So you might already be able to follow that if we understand the interaction between the abnormal cells and the normal cells, we should be able to identify a language, a signature that is actually characteristic of the type of interaction that helps these cells become fully blown malignant cells. We can develop not only the tests potentially that will help us make that accurate diagnosis, but also find a way to target the vulnerable areas where we can basically potentially cut off the support of the stromal system because we know which genes are involved, we know which cells are involved, and hopefully prevent the precancer from becoming that very serious problem.
We know current approach, surgery, chemo, radiotherapy, it's very harsh physiologically and psychologically. it's a lot of stress for patients. If we can find a way for early diagnosis, prevention, minimizing the physical and psychological stress, I think that's even a better one.
When community becomes involved in this significant way, it tells us people do care about this. People want us to do the work and find answers to these important questions. So we can't thank Paceline enough for being so positive and so supportive.